Okay, so we're going to start talking about microbial growth. The whole chapter is divided, the whole unit is divided in three kind of subchapters. General considerations for growth, chemicals for microbial, chemical and physical methods of microbial control, and then antimicrobial drugs. Okay? So I have very ambitious plans to do all of that stuff this week. So your next exam is going to be next Tuesday. Okay? Now, let's start with the discussion of the microbial growth. Kind of general considerations. Bacteria, we're going to talk about bacteria exclusively. They divide via binary fission. In a very broad strokes, DNA gets replicated. Cell enlarges. Cleavage furrow is formed. And you end up with two cells. Interestingly, the protein FTSZ, it's actually the whole family of proteins, FTSZ, that form the cleavage furrow. In terms of its properties, it's quite similar to protein actin in human cells. So it can contract and separate two cells, right? Now let's take a look at this whole process of division. So you start, let's say for simplicity, you start with one cell. Okay? Now, in the first generation, when one cell divides, you're going to have two cells. Right? When each of these two cells divides, the second generation will have 4 cells. Third generation will have 8 cells. Fourth generation will have 16 cells. Fifth generation is going to have 32 cells. Does that make sense? Now you can clearly see that those are exponents of 2. 2 squares, 2 cubed, 2 to the power of 4, and 2 to the power of 5th. Okay? And you can move on and on and on. Does that make sense? Couple of terms here. First of all, the time that it takes for a bacterial cell to divide is called a generation time. Bless you. Now, generation time is different between the laboratory and natural conditions. Okay, now I have a really good question for you. What do you think if I'll take a cell, let's say E. coli, can put it and we'll start grow it on the agar plate, versus same E. coli growing in the guts of a human? In which conditions the generation time will be shorter, meaning that the cell reproduces faster?
In the lab.
Well, why in the lab?
Well, it's not flooding anything.
Yeah. It's actually, yeah, you're absolutely right. There's no competition. There's abundance of resources. So for every microorganism, the generation time in the lab is shorter than generation time in nature. Does that make sense? Another thing that I want to highlight here is that generation time of cells greatly depends on things like temperature, and so we can put here, we can put this sign, you know. And generation time obviously depends on things like temperature, pH, oxygen concentration, etc. Am I clear? So things, along with this, one thing that you should be able to do is to calculate the generation time based on like this data. So for instance, let me explain. I am telling you that for some kind of bacteria, the amount of time that it takes to go from one cell to 32 is 1 hour. Are you following me? So you calculate, you say, okay, from 1 to 32 cells, it's 5 generations. Can you see that? It's 5 generations, which means this cell goes through 5 generations over an hour. So what's the generation time? 60 minutes divided by 5, so 12 minutes. Am I clear? Does that make sense? Good. Another thing that I can ask you is to do kind of a reverse. I can tell you, okay, we have 100 cells. Generation time is 30 minutes. How many cells are you going to have in 2 hours? Okay, so we say, okay, 30 minutes, generation time. 2 hours, 4 generations. We start with 100 cells. After 4 generations, we're going to have 1,600 cells. Am I clear? If you're curious whether or not you can use a calculator, you absolutely can. Yes. Does that make sense to you all? Good? Now, if you have, say you have a generation time for a cell at 30 minutes. You put one cell on the agar plate, you stick it in the incubator. Cell starts to reproduce in the lab. So in 24 hours it will go through 12 generations. In 24 hours it will go through 48 generations, 30 minutes, right? In 48 it will be 96 generations. I cannot even fathom what 2 in the power of 96 is. It's a really, really big number. So why the hell do we not have bacteria just spilling over the edge of the agar plate and crawling out of the incubator? What stops bacteria from that constant reproduction? Yeah, lack of resources, they run out of food. Does that make sense? And this brings us to another important discussion here. Discussion about the growth curve. So this is how we describe the growth of microorganisms using so-called growth curve. So growth curve is logarithmic. So it's convenient because think of it this way. I'm not gonna write some stuff. I'm gonna just tell you. So look. If you try to plot the growth of microorganism against time, okay, you will have to plot the first, second, third, fourth, fifth generation, and so on and so forth. You following me? By 10th generation, you're talking about 1,024 cells. By 20th generation, you're talking about 1,048,000 cells. So you need to put a number 1 million and a number 2 on the same plot, which makes it really hard to read. Does that make sense? So instead what we don't put the actual number of cells, we put a logarithm of that number. Okay? So here you're going to have logarithm of the number of cells, and on the on the x-axis you're going to have time. The convenience of logarithmic representation, strictly speaking semi-logarithmic, is that the parts of your the growth dynamic becomes linear on the graph. So let me show you what I mean. The first part of the growth curve is called a lag phase. During lag phase, nothing really happens. The microorganisms do not reproduce because they experience some stress due to the transfer to the new environment. Then when they have settled, they enter the logarithmic growth, the explosive growth phase, which in this semi-logarithmic representation looks basically like a straight line. So the second step is log or exponential or growth phase. And this part of the graph, during this period of the existence of the community, the amount of nutrients is much higher than the amount of wastes. And the rate of replication, number of replicating cells, way more than the number of dead cells. Does that make sense? Good? Over time, nutrients start to run kind of, not really low, but they start to decline. Wastes starts to climb, and the system enters the stationary phase. So during stationary phase, you still have quite a decent amount of nutrients, but the number of live cells is pretty much equal to the number of dead cells. Eventually, the amount of nutrients becomes really, really low, and the whole system enters the last decline or death phase. During that part of the life of the community, nutrients run really low. And number of live cells is much lower than the number of dead cells. Does that make sense? So, one question. Natural population of microorganisms, like the ones that live in your gut or on your skin, in which phase do they exist? Yeah, stationary, of course, because otherwise it would explode, right? Now, an interesting feature of the decline phase is that some microorganisms can form so-called persister cells during that step. Persister cells slow down their metabolism, which allows them to survive, the times when nutrients are sparse and wastes are abundant. Does that make sense? So actually, the accumulation of waste explains, some interesting features. If you ever, looked at beer or wine, you can reach certain limit on the alcohol concentration in those products. For wine, it's about 14%. For beer, it's probably about 14% as well, but that's like really, really strong one, okay? So what happens during fermentation, alcohol is essentially a byproduct of waste that microorganisms produce. And we all know that alcohol is toxic for microorganisms, right? So basically, the accumulation of toxic waste limits the growth and limits the concentration of alcohol that we can achieve in the fermentation products. Does that make sense? Now, in some applications, for instance, if we try to generate more and more butanol for engines, we want microorganisms to be constantly in a stationary phase. This can be done by using so-called chemostats, which my understanding of technology, it really is overwhelming. From what I understand, it's a big tank where you have nutrients going in and wastes going out. That's all. Does that make sense? Okay, we're good? Any questions on this one? All right.